Hello, Biology 107 fans. I am going to start right at the beginning of transcription, and where we started was the process of gene expression and looking at where this begins and how it starts. We know that transcription involves, this time we involve an RNA polymerase instead of a DNA polymerase. So DNA polymerase, we saw those, DNA Pol 1 and DNA Pol 3, those were used in replication. And now what we're going to do is we're going to talk about RNA polymerases and what they do with transcription. There are a lot of similarities, and then there are several key differences between the machinery that is used for replication and transcription, and those would be important points for you to try to understand for the purposes of taking an exam. We talked about the fact that we are now taking DNA sequence, which is DNA nucleotides, and they are going to be encoded for using RNA. And similar to what happened previously, the RNA polymerase can only work in the 5' to 3' direction. So this is the same. The DNA polymerase can only work in the 5' to 3' direction, and the RNA polymerase can only work in the same direction. Huge difference. So this is difference. The RNA polymerase does not require a primer. What that means is, just as the RNA primase was able to put down a small sequence of RNA to start the process of DNA replication, in this case, the RNA polymerase can start its transcription process without having a primer. It doesn't need a 3' hydroxyl. And we talked about why this is. It's because the enzyme itself doesn't require that particular shape to fit in, right? Remember the lock and key with the enzyme. The key doesn't require a 3' hydroxyl in the case of RNA polymerase. The RNA polymerase will make phosphodiester bonds. This is the same as what DNA polymerases do. In this case, the phosphodiester bonds are between RNA nucleotides. And the RNA polymerase will make an RNA molecule that is complementary to the gene. And this is, right, the same as DNA polymerase. It makes a strand that's complementary to the other strand. In this case, we're only looking at genes. And the gene will include the, in the case of the RNA, the 5' UTR, the coding sequence, and the 3' UTR. In bacteria, transcription, right, occurs, and this is important for converting genetic information into proteins. And this also occurs in eukaryotes as well. Now, this is where we got stuck the other day. We were talking about the RNA polymerase. It makes an RNA transcript. So the RNA that gets made is called a transcript. And remember which kinds of RNA we could make. We can make tRNAs, and those are important for the process of translation. We can make rRNAs. That's important for ribosomes. And we can make mRNA. And mRNA encodes for proteins. And then there are many other RNAs. There's snRNA, snORNA, siRNA. There's a lot of different RNAs, but the main ones that we talk about are these three: tRNA, rRNA, and mRNA. And the template strand, the strand that is going to be transcribed, so it's the template for making the RNA, is antisense. And the non-template strand, the strand that actually has in the DNA a T, G in the 5' to 3' direction is the sense strand. And the sense, it's called sense because you can read the sense of it. It will tell you the sequence, the codons that will be in the gene that you're encoding for. And this template strand is antisense because it's actually the complementary sequence from what is going to be made in the RNA transcript. The polymerase is going to once again do many of the similar things that had to happen during replication. DNA is going to be in its helix, and it has to be open, so you're going to have some sort of molecule that acts as a helicase. All right. Then you're going to have to polymerize the new strand. So on the bottom here, they're showing you the DNA template. And they are synthesizing the RNA strand here by complementary base pairing to the DNA strand. So you pair a C with a G, a C with a G, a U with the A, another A here is going to come in with the T, and what we're doing is we're using the energy from the incoming triphosphate, so in this case this is a ATP, to generate the phosphodiester bond that links, right, this is a phosphodiester bond that links the nucleotides together. The hydrogen bonding forms between complementary base pairs with 3 hydrogen bonds between C and G and 2 hydrogen bonds between A and U. In bacteria, the enzyme that does this is called the RNA polymerase, the hollow enzyme. And then we already talked a little bit about this. It's composed of 2 different subunits. The sigma subunit, and this is going to be really important for initiation of transcription, and the core enzyme that actually has the ability to generate phosphodiester bonds. Now, the sigma subunit, as we previously mentioned, there are many different sigma subunits that can interact with the core enzyme. And that's important for different cells, different types of cells, and how it's determined which sigma factor is going to be turned on is dependent on activities that occur in the cell. So the sigma subunit binds to the core enzyme, and then this allows for the sigma factor to find the start site for transcription. Now, be careful, because it's the start site of transcription, not the start site of what that gene ultimately is going to make. So it is not an ATG site or an AUG site. It is not either of those. It is the 5' UTR that it is going to recognize. And what it actually recognizes is the promoter sequence. We had begun looking at this image, and really all you need to know is in this one picture. The promoter sequence is composed of 2 different boxes. The -10 box is called the Tata box, and the -35 box is the -35 box. And these are upstream -10, so this would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right, maybe I didn't do that quite right. Let me erase that. Let's do it again. Okay. Because this is -10 right here. So this is 10 for the A, the T, I'm sorry. 9, 8, 7, 6, 5, 4, 3, 2, 1. Okay, and there is no 0. Okay, so it's minus 10 bases upstream of the transcription start site, which remember is going to include the 5' UTR. Minus 35 is a second box that's also recognized in the DNA sequence upstream, and this is also part of the promoter and recognized by the sigma factor in the holoenzyme. Both the Tata box here and the minus 35 box bind to the sigma factor and enable the DNA that's wound around to become opened. And what's nice about this is the sigma factor localizes the core enzyme. And the core enzyme has multiple activities. One of the activities is that it has a portion of its structure that acts as a helicase. And what it does is this allows the DNA that's threaded through the sigma factor to become unwound. And we need to do this, so this acts as a helicase, to allow transcription to begin. You see, one strand is separated and comes this way, and the other strand goes around that, it's actually called a rudder, in the other direction, and that opens up the DNA here to allow RNA transcription to begin. Notice they're going to call, there's two different parts of this, the zipper and the rudder. I don't really care if you call it rudder and zipper, right? But this is part of the holoenzyme. So this acts, the zipper and the rudder acts as the helicase. Now, as the DNA continues to be threaded through here, it's going to continue to open up the DNA. RNA is going to be polymerized using the enzyme binding site, and that's going to lead to the formation of an RNA transcript. Okay, so you start with initiation. Initiation is it started by the sigma factor that localizes the RNA polymerase to the right place. You are going to begin transcription, and that's really regulated by the polymerase and the rudder and zipper. And that's going to allow for elongation to occur. That is, as the RNA moves moves along the DNA template, you're going to be making RNA in the 5' to 3' direction. And then the last thing that has to happen is it has to stop somewhere, and there's actually a signal that causes the RNA polymerase to fall off the template strand, and that's usually due to a hairpin structure that forms in the RNA. And so as the RNA is being made, it's going to be made, and then at some point it's going to generate this hairpin structure that's going to cause the whole RNA to bump off of the enzyme. And that terminates transcription. What's the big difference between transcription in eukaryotes and transcription in prokaryotes? Well, kind of huge. There's only one RNA polymerase in bacterial cells, and it does transcription of everything, whereas you have multiple RNA polymerases in eukaryotic cells. Now, it's kind of an easy thing to do here. It's easy to remember this. RNA polymerase 1 does rRNA. And you're going to see why I'm going to tell you this is easy. So rRNA found in ribosomes. RNA polymerase 2 does mRNAs, RM. And then RNA polymerase 3 in general does... Where are the tRNAs? Interesting. I thought RNA polymerase 3 did tRNAs. I do believe it does tRNAs. Unless that's something that's changed, and I'll have to look that up. So one of each of these will do the majority of the transcription found in eukaryotic cells. We do focus almost all of what we talk about on the RNAs that generate mRNAs, because those mRNAs are then going to be translated into proteins. And see that we have a whole lot of additional proteins that play a role, and these are called transcription factors. And what's nice about this is they just started calling them TF, transcription factor 2, for the fact that we're using RNA polymerase 2. And they named them A, B, C, D, E, F, G, etc. And so here what you need to do is you need to remember the sequence of the transcription factors that play a role in eukaryotic gene transcription. Now, in prokaryotic, it was really easy because we just had the sigma factor and the core enzyme. We didn't have a whole bunch of stuff. That was all we needed in prokaryotes. And what the sigma factor did was allowed us to find the initiation site. And then the core enzyme had in it a helicase. And it had the enzyme that was allowing us to do the transcription. And all of this now is going to be taken over by eukaryotic transcription factors. So the first thing that's going to recognize, okay, a Tata box, and don't worry about how many base pairs upstream. Just remember what happens for the bacterial. Bacterial is -10, -35. Those are what's important. But in eukaryotes, you're still going to have a transcription factor that recognizes a Tata box. And then we're going to go through some additional transcription factors that have some of the same roles that we saw that were played out with the bacterial transcription RNA. So in this picture, you see the RNA polymerase in yellow. TF2D is localized to the Tata box. You have some additional transcription factors, B, E, A, and really this is probably the most important because it determines where that RNA polymerase is going to sit. TF2D has a Tata binding protein as part of that TF2D complex. So it actually has a TBP, Tata binding protein, in TF2D. I think that might be enough for understanding the polymerases. Let's move on to understanding what actually happens with respect to the transcript that's being made. So the transcript is the product that's made by the RNA polymerase. So it's the product of RNA pol. Now in bacteria, it's really simple because the RNA polymerase in bacteria will start at a 5' UTR. Do its sequence till it gets to the gene, continue through the gene, and have a 3' UTR. Alright, the gene is just coding sequence, so this all codes for some portion of the protein that's being made. However, In eukaryotes, we have additional sequences that are part of the gene sequence that are called introns. And introns stand for intervening sequences. So they're actually sequences of RNA, that will not be translated into protein. And we have to remove introns from the gene prior to the RNA transcript being exported out of the nucleus. So one of the interesting discoveries early on was that the RNA transcripts that were made, the original RNA transcripts that were made, were really long. So you would have the 5' end here and the 3' end here, and even if it had these sequences upstream and downstream, the actual length of the gene was way longer than what the length of the protein product was at the end. And that's how they figured out that there were these introns. So the ones that are coding for being in the protein are called exons. So these are kept. So these are in the protein. Whereas the introns are those intervening sequences that I said had to be chopped out. Here's an image showing you what it looked like when they looked at DNA versus RNA. And in B, what you can see is that here's the single-stranded DNA, and it's base paired with the mRNA. So these are DNA here. And then the RNA is much shorter. And what this means is that the DNA has lots and lots of sequence associated with it, and so it's going to be, especially when you get into the gene sequences, it's going to be a sequence of introns and exons that don't all necessarily get put into the final protein. And these are of varying lengths, and we call these exons 1, 2, 3, 4, and 5. And you might get a protein that's made of exon 1, exon 2, and exon 5. Or you could get a protein made of exon 1, exon 2, exon 3, exon 5. Now they have to go in order. They can't skip out of order. But these enable us to make multiple isoforms of proteins from one RNA transcript. So in this one, the yellow is the RNA transcript. And you can see that it's much shorter because at this point, the introns have already been snipped out. And so this shows you a little nicer exons and introns. So this is the size of the gene in DNA. The original RNA transcript would look just like this. And then the RNA, after it's fully processed, so this is RNA, but this is after all of the splicing of introns has occurred, which means they've been spliced out. So this sequence is spliced out, this sequence is spliced out, this sequence, this sequence, this sequence. And what you're left with is only this red sequence that is put together as a full-length protein here. So this is the final RNA. So this would be the primary RNA, is going to be the same length as the DNA, but once you have processing of that RNA, when it only contains exons, that's the final RNA that will then be exported out of the nucleus. And so remember, primary RNA is going to have exons and introns, whereas the spliced final transcript is going to have only exons. Exons mean expressed, introns intervening. Now, as I said, there's a couple things that have to happen before our RNAs can get out of the nucleus. So in eukaryotes, it requires this RNA processing. So you'll see RNA processing. And it involves 3 steps. You need to put on a cap. It needs to have a tail, a 3' polyadenylated tail. And you have to do splicing. All of this is going to occur, so all of these steps occur in the nucleus. What is a 5' cap? A 5' cap is just that. It puts a little hat, right, on the end of the DNA, and it protects that end of the DNA. So it has 2 functions. So it protects, and it also aids in the process of translation. What about a 3' poly A tail? Well, that's going to help, right, in having an RNA that has a whole bunch of A's at the end, because if it starts to get degraded, if these start to come off, if you have approximately 200 A's, nothing bad is going to happen to that RNA until you chew all the way back through that poly A tail. So this serves as a protective device as well. So this poly A tail has to happen. This cap has to happen. And we have to do the splicing that gets rid of introns. I'm not going to talk too much about this. There is a specific series of proteins. Notice one of them here is an snRNP, which is a small nuclear ribonuclear protein. We did talk a little bit about these earlier, but it's a conglomeration of proteins that make a complex called the spliceosome. And the spliceosome takes big, huge chunks of introns and cuts them out, and then ligates the 3' end where it cut to the new 5' end. Okay, these loops are called lariats, just like a cowboy would swing a lariat and then rope a horse. This is called a lariat, and the lariat gets ligated together, and these will get degraded eventually in the cell. And this leaves us with the exons bound one to another. So splicing removes the introns, capping adds this 5', it actually has a name, it's a 5' methyl guanosine. Guanosine is a nucleotide, and it's actually a methylated nucleotide that gets turned upside down with its phosphate groups and slapped on top of the RNA. And then the polyadenylation is added to the end of the DNA. So pre-mRNA, that's what happens first. Then you have it processed, and then this pre-mRNA is exported as mRNA. So it gets exported as mRNA. In a eukaryotic cell, all of that processing occurs in the nucleus, and only mature mRNA gets transported out of the nucleus into the cytoplasm. In the cytoplasm, that's where the mRNA will meet up with a ribosome to initiate translation. If we're going to compare, okay, and this is thrown in here as a way for you guys to think about organizing this in your head. Okay, if you think about RNA polymerases, in bacteria there's only 1. In eukaryotes we have 3. We mostly talked about RNA polymerase II, which is the one that does mRNA synthesis. The promoter structure, right, has that -10 and -35. It has a Tata box and the -35. In eukaryotes, there's a Tata box and then another box. They have variable lengths of sequences upstream of the start site. The proteins involved for bacteria are the sigma proteins, and there's lots of different sigma proteins. And most importantly here, there's no RNA processing. It doesn't really happen at all in bacteria. And one nice thing about this is that since bacteria are doing this, they're generating, so they have their DNA, right? Their DNA is in the cytoplasm. They're making RNA right off of the DNA, and this RNA will be translated as it's being made. And so you get all, right, you get the protein made as the RNA is being made from the DNA. In eukaryotes, totally different story, right? Because we have to have, here in a eukaryote, you're going to have the DNA, which is in linear chromosomes, is going to get opened up to make the RNA. Okay, that RNA is going to get processed. So all of this occurs in the nucleus, and then out in the cytoplasm, we get the binding of ribosomes to generate the protein product from those RNA transcripts. Okay, this is another example of what we were talking about the other day. So remember, the template strand, this is going to be the template, because when it gets transcribed, all right, you're going to have the opposite orientation. So here's 5' to 3', 3' to 5', and C goes to G, G goes to C, blah, 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 blah. Eventually what you reach is this AUG, which is going to be, so this is going to be the start of translation. So this is going to be your 5' UTR. And what this means is that this is called the sense strand, because you could just read the sequence as this without doing anything more. So this is encoding for a methionine, this is coding for another amino acid, I don't know which amino acid, etc. And so you can read these in threes until you get to a stop. And notice that if you look at the template strand, okay, this here is the template strand, you don't see that ATG in the 5' to 3' direction. What do you see? Well, what does ATG come out to? It comes out to a TAC right here. All right, and really, if you want to look for something, it's cat in the 5' to 3' direction. So as we move along, right, you would be able to then translate this, right, using the codon chart to make a protein product. What happens if you do the transcription of this? Well, you get this product, all right, so 5 to 3, 3 to 5, and as I said, okay, here's your ATG. This doesn't contain an AUG sequence. It actually has, where is that? It's hard for me to see this as I'm doing it on the computer. So if we look for cat, okay, it's really not going to be cat, it's going to be CAU in the RNA. So, all right, remember if it's DNA it would be that. So in the DNA it's cat. So this doesn't encode for the protein. That's why it is called the non-template strand, or the sense strand. I know it's confusing. Okay, do the best you can. All right, template, antisense, non-template, sense. All right, hope that helps you guys out, and we'll continue from there on Wednesday.